Okay, hello everyone. I'm Hania Nizami, and I'm going to be giving a lecture on porphyrin and hemoglobin synthesis. As you can see, I reused the same slides as one of your lectures. I've just reordered them a little bit and added some extra pictures. And just so you guys know how this works, the way that I usually do this is that I will teach the concept first, usually using diagrams or something, and then I'm going to test you on it later on in the slides. So the way that you can do that is that you can just pause and try to answer the questions that are on the screen. So let's begin. So because the title of this lecture is Porphyrin and Hemoglobin Synthesis, let's first go over what porphyrin is. I feel like it's important to understand that. So over here on the left side of my screen, you can notice that we have a pyrrole structure, right? And so what happens is that if you attach 4 pyrrole rings, what I'm pointing at are pyrrole rings, do the same thing as the thing on this side, on the left side of the screen, and you attach them using methine bridges, you call this entire structure a porphyrin. So a porphyrin is made out of 4 pyrrole rings and is attached to each other by methine bridges. And what happens is that the middle of a porphyrin ring is actually slightly negative. And so what that means is that if I have is negative. And so what that means is that if I have a positive ion, such as the ferrous ion in this situation, which is Fe2+, the ferrous ion can go in the middle of the porphyrin and attach. So again, this is my porphyrin on the outside, and in the middle of my porphyrin, I can have a positive ion, in this case, ferrous. If ferrous ion is present, or the Fe2+ ion is present, we call this entire structure hemoglobin. So hemoglobin is a porphyrin that has Fe2+ in the middle. Okay, so I'm going to test you guys on what I just taught in the last one. So porphyrins are formed by the fusion of what rings? What are the name of the rings? Pyrrole rings, and they're linked by what kind of bridges? Methine bridges. And what is the name of the ion that's in the middle of the porphyrin ring in the case of heme? What is the name of that ion? Or it's Fe2+, which is iron, specifically ferrous iron, as you can see over here. So let's just go over some of the other types of porphyrin rings that we might encounter. As we talked about over here, we have heme. I can see my porphyrin ring. I can see Fe2+ in the middle. There's also porphyrins called chlorophyll A and chlorophyll B. These ones aren't found in humans. Instead, they're found in plants, and they're really important for photosynthesis. And in the middle of chlorophyll A or chlorophyll B is magnesium. Okay, so in the middle of chlorophyll A and chlorophyll B is magnesium. I'm going to test you on that in the next slide. And then lastly, if we look over here, I have my vitamin B12. This entire structure is my vitamin B12. And in this segment over here, I can recognize my porphyrin ring. I can see my pyrrole rings over here, and I can tell that they're connected by methine bridges. And in the middle of them, I can see my cobalt ion. So vitamin B12 is also called cobalamin. And the reason it gets that name is because in the middle of the porphyrin ring is cobalt. So vitamin B12 is made out of cobalt. Let's just go over those questions again, or over those concepts again. Chlorophyll, what's the name of the ion that's in the middle of it? Magnesium. Heme has what ion in the middle of it? Iron, specifically ferrous ion, Fe2+. And then cobalamin, what's in the middle of cobalamin? What's the name of that ion? Cobalt. And cobalamin is also called vitamin B12, and it's important for red blood cells and the health of nervous tissue. So like I talked about before, heme is made up of Fe2+. If you oxidize the Fe2+, then you can get Fe3+, because remember, oxidation is a loss of electron, aka getting more positive. So Fe3+ is the oxidized version of Fe2+, and we call this form of iron the ferric form of iron. And if the ferric form of iron, or Fe3+, is found in the middle of the porphyrin ring, then we call this hemin. It's no longer called heme, it's called hemin. So essentially the idea is that hemin is the oxidized form of heme. In some cases that we're going to see later on, sometimes instead of iron in the center, you're going to find zinc. And this is usually in cases where the iron is unavailable, either because the enzyme isn't working or because there's no iron to be found. So again, in human beings, sometimes if we don't have iron or if the iron can't be utilized, instead the body is going to replace Fe2 plus with zinc. And we call this Zinc-Protoporphyrin. The other thing that they want you to know is that usually when we talk about hemoglobin, the heme group that we're talking about is specifically heme B. So heme B is the specific form of heme that's found in hemoglobin. Heme A and heme C are instead found in cytochromes. And if you remember, cytochromes play an essential role in the electron transport chain that ultimately generates ATP. So again, it's important to know that the specific form of heme that's found in hemoglobin is heme B. And heme A and C are found in cytochromes, which play a role in the electron transport chain. So the other thing that they want you to know about, I think the main thing that they want you to understand is that heme is not just found in hemoglobin. It's found in a lot of other areas. One of the areas that it's found is called the cytochrome P450 enzymes. And the reason these enzymes are really important is because they play a large role in detoxifying substances, especially in the liver, such as like drugs and that kind of thing. So the main idea over here is that cytochrome P450 contains heme. This is going to be important because later on, I'm going to talk about how this can actually affect hemoglobin. So cytochrome P450 contains heme. That's the main thing that I want to get out. And that there are some other structures in our body that also contain heme. Obviously, we know hemoglobin, and we know that its role is oxygen transport, right? Versus myoglobin, which also contains heme, but this time it's oxygen storage in muscles, right? Because when you're exercising, you need more oxygen than normal. And so you can store that oxygen in the form of myoglobin. We already talked about cytochromes. Cytochromes specifically have heme A and heme C, and they play a role in the electron transport chain that ultimately results in the formation of ATP. Heme can also be found in catalase. Catalase is an important enzyme that our body uses to break down bacteria. And then heme is also found in the enzyme nitric oxide synthase. So again, I don't think you really need to spend a lot of time memorizing these. The main idea is that heme is found in more places than just hemoglobin. And one of the places that it can be found is the cytochrome P450. So I'm sure you guys already know about the structure of hemoglobin, that it has two subunits. Most of the time it's two alpha, two beta subunits. The main thing that I want to test you guys on is that what kind of heme do we find inside hemoglobin? The answer is heme B. Heme B is found inside hemoglobin. And what do you call this version of iron that you find inside hemoglobin? What type of iron is this? The answer is ferrous iron. Fe2 plus is called ferrous iron. So these are some points that I believe you guys already have learned in your other lectures, but red blood cells have a lifespan of 120 days, which means that they're going to renew themselves approximately every 3 to 4 months. Obviously, when you're making hemoglobin, because heme is made out of porphyrins, you will need to also synthesize more porphyrins when you want to make more hemoglobin. The other idea they want you to know is that bound iron is recycled. So once you break down one RBC, you can remove the iron and you can actually use that same iron, bring it back down to the bone marrow, and now we can use it to make new RBCs. So it's actually the same iron that's being reused in your body. And then I'm sure you guys know this, but one of the important functions of the spleen is to phagocytose RBCs once they've reached the end of their lifespan, or sometimes also in cases if the RBCs are malformed. So the role of the spleen is to phagocytose RBCs either when they've reached the end of their lifespan, or also in cases where they're malformed. So this is just a really easy review slide. What is the name of the heme-containing compound that's responsible for oxygen transport? Hemoglobin. What's the name for the one that's responsible for oxygen storage? Myoglobin. Responsible for electron transport? Cytochromes. Remember, all of these contain heme. We already talked about how the cytochrome P450 enzyme also contains heme, and this is going to come up, it's going to be important later. You guys remember I talked about an enzyme that's important for the breakdown of bacteria that also contains heme. It's called catalase. Okay, so the things that I talked about before were a good revision, but now we're going to really talk about the most important part of this lecture. If you walk away with one thing from this lecture, it should be that you memorized this slide, because this slide is very, very, very heavily talked about and heavily tested about. So I'm going to continuously repeat the ideas from over here. So what we're talking about right now is we're talking about the synthesis of heme. So this oval structure over here is my mitochondria. So just take a moment to appreciate that we have some of the process of heme is occurring inside the mitochondria, specifically the beginning of the process and the end of the process. And the middle of the process is actually occurring in my cytosol. So now let's start with the actual process of heme formation. It starts with succinyl-CoA and glycine, and they're going to react together to form this structure over here, which is called ALA, or delta ALA. And it's not going to surprise you, but the enzyme that's responsible for making ALA or synthesizing ALA is called ALA synthase. So I know that these enzymes seem really intimidating when you first look at them, but it should be really easy when you break down the name of these. ALA is synthesized from ALA synthase. That's an important thing to remember. So this is the very first step in my heme formation. The other thing you really want to remember is that I put the star in this step because this is the rate limiting step. A lot of the times they really like to ask you about what is the rate limiting step in heme formation. And your answer should be the very first step, which contains the enzyme ALA synthase. ALA synthase is the rate limiting enzyme in heme formation. That's really important to know. The last thing I want you to know about this very first step is that it requires the cofactor vitamin B6. All right, so sometimes what they'll try to tell you is that there's a deficiency in vitamin B6, and they're going to ask you what step of the pathway can't perform. And the answer is it's the very first step, because the enzyme ALA synthase requires the enzyme vitamin B6. The other way that they might test you on this is telling you that ALA synthase has a necessary cofactor. Which cofactor is it? Vitamin B6. And if you don't have vitamin B6, that means that you can't form ALA, because ALA synthase will not be able to work. Okay, now that we have that under control, let's look at this next part over here, which is when my ALA is actually going to leave the mitochondria and it's going to go to the cytosol. So over here, you can see my delta ALA is in my cytosol. Essentially what happens is that two delta ALA are going to combine, and they're going to condense actually, and they're going to form what we call PBG, or porphobilinogen. I'm going to reference this as PBG for the rest of the lecture. So again, two ALA molecules are going to combine, and they're going to form porphobilinogen, or PBG. Okay, and the enzyme, again, this shouldn't surprise you, but ALA is going to be dehydrated. Dehydrated means we're removing a water molecule by the enzyme ALA dehydratase. So again, it shouldn't surprise you that the first enzyme is ALA synthase. First, I need to make my ALA molecule, and then the next step, I need to metabolize it using ALA dehydratase. Once I remove a water molecule, now my ALA has turned into porphobilinogen, also known as PBG. Okay, so the second step revolves around ALA dehydratase. It converts ALA to PBG. And the other thing that's really, really, really important for you to know, and this is very heavily tested, is that this step of the pathway can be inhibited by lead. Lead can inhibit ALA dehydratase. And the way that they might test you on this is they might ask you, what is the urine analysis that you might be expected to see in lead toxicity? right? And if you know that lead inhibits ALA dehydratase, then you should be able to figure out that because I'm inhibiting this enzyme, the substrate of this enzyme is going to accumulate, meaning it's going to be in very high quantities, which means that I'm going to have very high levels of ALA, right? Because ALA is my substrate. And on the other side, I'm going to have very low levels of PBG because I'm inhibiting the enzyme that's responsible for the formation of PBG. Okay, so lead poisoning is going to lead to high levels of ALA and low levels of PBG because it inhibits the enzyme ALA dehydratase. Okay, so now let's move on to the second one. In this next step, it involves the enzyme uroporphobilinogen synthase. So again, it should not surprise you by the name of this, uroporphyrin synthase synthesizes uroporphyrin. This shouldn't surprise you. So again, the next step or the next enzyme that's responsible for this step right over here is called uroporphyrin synthase, and it's responsible for making either uroporphyrin 3 or uroporphyrin 1. And the thing that I really, really want you to know, and again, this is very, very, very heavily tested, is that an enzyme deficiency in uroporphyrin synthase is going to lead to a condition, an inherited condition called acute intermittent porphyria. Okay, yeah, so uroporphyrin synthase is the next step. Is the next enzyme in the pathway. It makes either uroporphyrin 3 or uroporphyrin 1. And a deficiency in this enzyme can lead to a condition called acute intermittent porphyria. So the way that they're going to test you on this is that they're going to give you a clinical scenario of somebody with acute intermittent porphyria. Usually the symptoms of acute intermittent porphyria are either neurological symptoms, so something like weakness, paralysis, anxiety. Remember, anxiety is also a neurological problem, or something like abdominal pain. So they're going to give you a scenario. Somebody's presenting with acute intermittent porphyria. Another hint that they might give you for that it's acute intermittent porphyria is that there's dark colored urine or wine colored urine. And they're going to ask you what enzyme is deficient. And so you should know that it's uroporphyrin synthase. Okay, and the other way that they might ask you this, which is going to be a little bit harder, is they might ask you what levels do you think are going to be low, or what levels do you think are going to be high? And then the answer is that you're going to expect high levels of PBG, because once again, this is the substrate for the enzyme, and low levels of uroporphyrin if you lack the enzyme uroporphyrin synthase, as in the case of acute intermittent porphyria. So now let's move on to the next step in the pathway. This to convert uroporphyrin 3 or uroporphyrin 1 into co-proporphyrin, you need the enzyme uroporphyrin decarboxylase. So again, I know the names of these enzymes seem really intimidating when you first see them, but just think about this logically. I'm trying to metabolize uroporphyrin. And so if I see an option such as uroporphyrin decarboxylase, that's telling me that I'm removing carbon from uroporphyrin, which means I'm metabolizing uroporphyrin, and I'm converting it to this molecule right over here, which is called co-proporphyrin 3. So once again, the enzyme that's responsible for this is uroporphyrin decarboxylase. And the reason that I'm really emphasizing on this is that once again, you need to know a condition that is associated with this, an inherited condition that is associated with this, and it's called Porphyria cutaneous tarda. Porphyria cutaneous tarda is a deficiency in the enzyme uroporphyrin decarboxylase. And now that you know that, you should be able to recognize that it's going to lead to elevated levels of uroporphyrin, because once again, this is the substrate, and decreased levels of co-proporphyrin. So again, the way that they might present this to you is they're going to give you a clinical scenario of Porphyria cutaneous tarda. And if you look at the name of this disorder, you'll notice that the middle thing has Cutanea in it. Cutanea means skin. And so if you see a clinical scenario and they're telling you a scenario of skin manifestations, specifically photosensitivity, which means that the patient might have gone out in the sunlight and they got a lot of blisters afterwards. The first thing that should be popping to your head is Porphyria cutaneous tarda. So now that you've established that it's Porphyria cutaneous tarda, because you're seeing somebody that's went into the sunlight and they had some skin manifestations, the second thing that's popping into your head is that I know that this is a deficiency in the enzyme uroporphinogen decarboxylase. And because you're aware that it's a deficiency in the enzyme uroporphinogen decarboxylase, you already know that it's going to lead to high levels of uroporphinogen and low levels of proporphinogen. So that's the main things and the main ways that they might test you on this. The next two steps in this pathway aren't incredibly important, but essentially the coproporphinogen is going to go inside the mitochondria. It's going to be metabolized into protoporphinogen 9, and then the protoporphinogen 9 is going to be metabolized into protoporphyrin 9. And then the protoporphyrin 9 is going to form heme. Now, the way that this happens is that I have my porphyrin, right? I have my protoporphyrin 9, and what I need to add to make it into heme is iron, right? In the middle of this porphyrin, I need to add an iron, specifically ferrous iron Fe2+. And the way that this happens is using the enzyme ferrochelatase. So the very last enzyme in the heme synthesis pathway is ferrochelatase, and it's responsible for adding Fe2+ to porphyrin. Okay, and once again, a very, 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 very high yield topic that you guys should know is that lead can also inhibit this enzyme. So lead does not only inhibit ALA dehydratase, lead also inhibits ferrochelatase. Okay, so this is really important because lead is going to decrease the activity of ferrochelatase, and that means that iron cannot be incorporated into protoporphyrin 9, and therefore I can't form heme. Because I can't form heme, I can't form hemoglobin, and that's why you get anemia and lead toxicity. Okay, so lead inhibits two enzymes. It inhibits this enzyme over here, ALA dehydratase, and it's also going to inhibit this enzyme over here called ferrochelatase. Okay, this is really important. Okay, so I'm just going to revise everything that we've just done. I'm going to go over it again, and then the rest of this lecture, I'll be actually quizzing you on this. So let's go over the first step in the pathway. We talked about how we have glycine, we have succinyl-CoA. They're going to combine together using the enzyme ALA synthase. ALA synthase is the very first enzyme, and it's also the rate-limiting enzyme. This is very important, very high yield. They're going to ask you what is the rate-limiting enzyme in heme synthesis. It's ALA synthase. And the other thing they really want you to know is that vitamin B6 is an essential cofactor for ALA synthase. What is the essential cofactor for ALA synthase? It's vitamin B6. Okay, so now that I have my ALA molecules, what's going to happen in the next step, or the second step, is that my ALA is going to leave the mitochondria, it's going to go into the cytosol, and two of the ALA molecules are going to attach together, and using the enzyme ALA dehydratase, it's going to form a molecule of porphobilinogen, which is also called PBG. So again, two ALA molecules combined together to form PBG using the enzyme ALA dehydratase. It's called dehydratase because it removes a water molecule or two water molecules. And the other thing, remember, that I really emphasize about this enzyme, ALA dehydratase, is that it can be inhibited by lead. So it's really, really important to know, because they're going to give you a scenario. Maybe a lot of the time that they're going to give it to you is that the child might have been eating toys, and a lot of toys actually contain lead in them. And the other scenario that they might be giving you is something to do with paint. It's a really old house, and that they're painting the house, and paint also creates lead. And so what's going to happen is that now that you have those buzzwords, the first thing that's popping into your head right now is that, oh, okay, I'm thinking of lead toxicity. And so the symptoms of lead toxicity that we're going to go over later are stuff like lethargy, fatigue, abdominal pain, things that aren't really specific. But you know that lead is found in toys, and you know it's found in paint. So already at the top of your head, you're thinking lead. And the other thing that you know is that lead inhibits the enzyme ALA dehydratase. Because you know that, you also know what's going to be able to be found in the urine. You know that there's going to be very high levels of ALA, but low levels of PBG. This is very characteristic of lead poisoning, high levels of ALA and low levels of PBG, which makes sense because I'm inhibiting the enzyme ALA dehydratase. Okay, the next step is that now that I have PBG, you're actually going to take 4 molecules of PBG. You're going to use the enzyme uroporphinogen synthase in order to make uroporphinogen. Again, from the name of the enzyme, uroporphinogen synthase synthesizes uroporphinogen, right? But the main thing that you should know about this is that a deficiency in this enzyme, uroporphinogen synthase, can lead to an inherited condition called acute intermittent porphyria. And let's just revise once again, what are the symptoms of acute intermittent porphyria? It's mostly neurological symptoms. So motor problems, weakness problems, anxiety, and it might also be something like abdominal discomfort, also something called wine urine. I'll go more specifically into that, but just keep in mind, if you hear those kind of buzzwords, you're thinking of acute intermittent porphyria. Acute intermittent porphyria is a deficiency in the enzyme uroporphinogen synthase. If I don't have the enzyme uroporphinogen synthase, I'm going to have low levels of uroporphinogen and high levels of PBG, because again, PBG is the substrate for uroporphinogen synthase. Okay, afterwards, after you have your uroporphinogen, you can decarboxylate the uroporphinogen to form coproporphinogen 3. And if you have a deficiency in this enzyme right over here, uroporphinogen decarboxylase, the condition is called Porphyria cutanea tarda. Porphyria cutanea tarda presents with symptoms, skin symptoms. Again, the huge hint is cutanea in the name. So specifically, it's going to give you a scenario of somebody that went out into the sun and they had blisters or something like that. Once you know that Porphyria cutanea tarda is a deficiency in uroporphinogen decarboxylase, you're also going to know that it's going to lead to low levels of coproporphinogen 3, but high levels of uroporphinogen 3, because the substrate of uroporphinogen decarboxylase is uroporphinogen, right? That makes sense. So you're going to have very high levels of uroporphinogen. Okay, the next 2 ones aren't super important, but after you form coproporphinogen 3, you can oxidize it using coproporphinogen oxidase. You get protoporphinogen 3, which you can oxidize using protoporphinogen oxidase. And then that's going to lead you with protoporphyrin 9. Just in case, I don't think this would come up in an MCQ, but in case it did, and they ask you about the order of the enzymes, the way that I would figure this out, in case you're getting mixed up with these two, is I would just look at the Roman numerals. So 3 comes before 9. So that would help me at least memorize that coproporphinogen 3 comes first before protoporphinogen 9. Just in case it comes up in an MCQ. But again, these 2 enzymes aren't as important. The main one is that after you form protoporphyrin 9, you need the enzyme ferrochelatase, which is going to add ferrous iron into the protoporphyrin 9 in order to create heme. This is the very last step in the heme pathway, and it can be inhibited by lead. The same way that ALA dehydratase in the previous slide could be inhibited by lead, ferrochelatase can also be inhibited by lead. So all the things I mentioned before about lead poisoning, where I talked about that it's going to be associated with either toys or paint, keep in mind that you're not only thinking of ALA dehydratase inhibition, you're also thinking of ferrochelatase inhibition. Okay, so just some other points to know about. The thing about red blood cells, obviously red blood cells carry hemoglobin. The thing about red blood cells is they actually don't have mitochondria. And as we've just revised in the synthesis, we know that in order to create heme, we need mitochondria. So the main thing they're trying to tell you is that hemoglobin actually isn't made inside of RBCs. Instead, it's made in other cells, such as erythrocyte cells, that are found, for example, in your bone marrow or in hepatocytes. But once the RBCs mature, it no longer contains a mitochondria. Because it no longer contains a mitochondria, it no longer contains hemoglobin. Okay, so now that I've gone over the most important things, the rest of this lecture is going to be a lot of quizzing you guys. So first, let's try to remember what was the name of the enzyme that was important for the committed step. The committed step is also called the rate limiting step. So what was the name of the enzyme that's involved in the rate limiting step of heme synthesis? Okay, the answer is ALA synthase. And my next question is, what is the name of the cofactor or coenzyme for ALA synthase? ALA synthase needs something. What is it? What is the name of it? Vitamin B6. Vitamin B6 is the name of the coenzyme for ALA synthase. Okay, so this next thing that I'm talking about is a little bit of a new concept, but I'm sure you guys are already well aware of it. So let's just think logically for a second. We're talking about heme synthesis, right? Which means that the very end product of this pathway is heme, right? And so just think for a second, if I have a lot of heme already, my body is already fully saturated on heme, do you think I still want activity of the ALA synthase enzyme? Do you think I still want to be creating more ALA, which is eventually going to lead to more heme? So think about that for a second and try to think, is heme and hemin, are they going to inhibit or are they going to promote ALA synthase transcription? Think about that. And the answer is that they're going to repress or they're going to inhibit the transcription of ALA synthase. So again, this should make sense. It's just basic negative feedback inhibition that we've always learned about. The end products of this pathway, such as heme or hemin, can actually repress the enzymes in the pathway, such as ALA synthase in this case, because you want to stop making the formation of heme because you already have enough of it. Okay, and then something else just to note is that it's an allosteric inhibition. This should make sense to you because the two types of inhibition are either allosteric or you can do competitive inhibition, right? It wouldn't make sense if this was competitive inhibition because ALA looks nothing like heme. We already saw how many transformations it has to go through until it starts to look like heme. And remember that competitive inhibition only works if the two molecules look the same. So instead it's allosteric inhibition. Allosteric inhibition is essentially when the ALA is going to attach to some part of the enzyme that's not the active site. It's going to change the shape of the enzyme. And because it's changing the shape of the enzyme can't really work as much anymore. The other thing that you want to know about is that there are certain things such as aromatic drugs. Aromatic drugs are specifically, like usually like anti-epileptic drugs, xenobiotics, and steroids. They can actually induce the synthesis of ALA. And the reason that this is bad in some scenarios is that if you already have a porphyria, if you already have an enzyme deficiency and you're increasing your ALA synthase, what this means is that it's going to increase the amount of substrates even more. And if you're increasing the amount of substrates even more, it's going to lead to an exacerbation of the disease. An exacerbation pretty much means that the patient's going to come in and they're going to have a lot of symptoms all of a sudden. So yeah, it's going to lead to an accumulation of the substrates in the scenario that you have an enzyme deficiency. And so the main thing that you want to look out for is that if they mention someone who has one of the porphyrias that we've mentioned, either acute intermittent porphyria or porphyria cutaneous tarda, and you give them aromatic drugs, xenobiotics, or steroids, it's actually going to make the disease even worse because these three things over here, induce the synthesis of ALA synthase. Because you're inducing the synthesis of ALA synthase, it leads to more substrates. More substrates is going to lead to more disease severity. Okay, so let's continue quizzing. So what's the name of the enzyme that's responsible for incorporating ferrous iron into the protoporphyrin? What is the name of that enzyme? The answer is ferrochelatase. And my second thing is that the thing that we just talked about in the previous slide, what is an example of a feedback inhibitor of ALA synthase? Negative feedback inhibitor of ALA synthase. Heme and also sometimes hemin, which is the oxidized form of heme. Okay. And the next question is that how do certain drugs, such as aromatic drugs or xenobiotics and steroids, how do they affect the rate limiting enzyme? Do they increase the production of it or do they decrease the production of it? The answer is increased production of the rate limiting enzyme. Because they do that, you get more substrates. Because you have more substrate, it's going to lead to an exacerbation of the disease if the patient has a porphyria. And my second question is, what is the name of the rate limiting enzyme? ALA synthase, right? That's really important. Also remember that ALA synthase requires a coenzyme vitamin B6. And then we talked about this, but hemin is the ferric form of, is the oxidized form of heme, and it's essentially going to decrease the activity of ALA synthase, right? So what do you call that? It's called feedback. Is it inhibition or is it promotion? It's inhibitor, right? Hemin is a feedback inhibitor of ALA synthase because it is the end product. And so through feedback inhibition, it would lead to decreased activity of ALA synthase. Sometimes what they want you to know is how exactly it decreases the activity. So one thing that we talked about is the feedback inhibitor. Remember, I talked about that it can bind to the enzyme, change the shape of the enzyme, so the enzyme can't work as well. That's called feedback inhibitor, right? Specifically allosteric. But it actually has three things that it can do. So first thing is feedback inhibitor. Second thing is that it can also decrease the transport of ALA synthase. And the third thing is that it can also decrease the synthesis on a gene level of ALA synthase. So one is the activity. I'm changing the shape of the enzyme. That's the first thing. The second thing is that I'm decreasing the transport. And the fourth thing is that I'm decreasing the synthesis. So sometimes what they'll do is that they'll give you an MCQ and they'll ask you, how does hemin inhibit ALA synthase? And you should choose one of these options. Either that's a feedback inhibitor, that it decreases the transport, or it decreases the synthesis. So it interferes with the DNA process. And the last thing that they just want you to know that, like I talked about before, hemin is the oxidized form of heme. And so sometimes you can see that in cases of vascular injury or hemolysis. Because it's the oxidized form of heme, it contains the oxidized form of iron, which is Fe3+. Okay, so ALA dehydratase, remember this is the second enzyme in the pathway. What is it inhibited by? What is the name? What is the thing that can be inhibited by? Lead, right? And also, what is the second enzyme that can also be inhibited by lead? What was the name of that enzyme? Ferrochelatase. The very last enzyme in the pathway, ferrochelatase, can also be inhibited by lead. Okay, so like I talked about before, one of the very characteristic findings of lead poisoning is increase in ALA, but a decrease in PBG. And remember, this makes sense if you can remember that lead inhibits the enzyme ALA dehydratase. ALA dehydratase is responsible for converting ALA to PBG. And so if I can't convert ALA to PBG, what happens is that the ALA accumulates. So I get very high levels and I get low levels of PBG, right? And then the other thing that they want you to be aware about is the fact that in both iron deficiency and also lead poisoning, you can get the formation of something called Zinc-Protoporphyrin. And so let's just think about this for a second, because I talked about this before. If I have my protoporphyrin and I can't incorporate the iron into it, either because there is no iron in my body or because the ferrochelatase enzyme is being inhibited, and so I can't incorporate the iron, what's going to happen instead is that zinc is going to replace the iron, and that's going to lead to the formation of Zinc-Protoporphyrin. Okay, so just it makes sense logically if you think about it. If I have iron, but I can't incorporate it, as in the case of lead poisoning, or I don't have iron, that means that instead of iron in the middle of the protoporphyrin, I'm going to have zinc. That's going to lead to Zinc-Protoporphyrin. Now, the next question is, okay, both of these cases lead to Zinc-Protoporphyrin. So now how do I tell the difference between the two? How do I tell the difference between iron deficiency and lead poisoning? And the answer is that there's these things that you can see inside the RBCs that are essentially hemosiderin particles. And hemosiderin particles are actually iron. So I'm going to show you a picture later on, but you'll find an RBC and you'll find these blue stained dots. Those blue stained dots are called hemosiderin, and hemosiderin is the accumulation of iron. Okay, so just think about this logically. If I have an accumulation of iron, would that be indication of iron deficiency, or would that be indication of lead poisoning? It has to be in the case of lead poisoning, right? It doesn't make sense that I have an accumulation of iron and iron deficiency. That doesn't make sense. So what happens is that in lead poisoning, I'm inhibiting the ferrochelatase enzyme. Because I'm inhibiting this enzyme, I can't incorporate the iron into my porphyrin. And because I can't incorporate the iron, it's just staying inside of my RBC or inside of my erythroid cell or my hepatocyte or whatever. And it's accumulating, and because it's accumulating, you can actually stain it, and you call that stain, or you call those particles, hemosiderin. So again, an accumulation of iron can only be seen in lead poisoning, not in iron deficiency. And you call that accumulation of iron hemosiderin. Okay, so what are two scenarios that the iron can't be incorporated into protoporphyrin 9? Or what are two scenarios that would lead to the formation of zinc-protoporphyrin that I just talked about? The answer is lead poisoning and iron deficiency, right? I can't incorporate the iron. Instead, what my body's going to do is incorporate zinc. It's going to lead to the formation of zinc-protoporphyrin. And they're just letting you know that this zinc-protoporphyrin is very easily detected. Okay, so this is just a little bit of a fun fact, I guess. But if you see something as ogen, that means that it's colorless. And if you see something that ends in, it means that it's actually very highly colored. And you guys actually know about this because you know about bilinogen, for example. I think you guys, I don't know if you guys took GI yet. But ogen just means colorless. And then if it has the suffix in, it means that it's colored. Okay, so we've already kind of touched on this, but I just wanted you guys to know that the two porphyrias that we talked about, acquired intermittent porphyria and the second porphyria, which is Porphyria cutaneous tarda, both of those have an autosomal dominant pattern. So sometimes it'll come up in an MCQ, what is the pattern of inheritance for acute intermittent porphyria or Porphyria cutaneous tarda? And the answer is autosomal dominant. So I am going to be testing you on this later. The only porphyria that is recessive is congenital erythropoietic porphyria. The only porphyria that is recessive, autosomal recessive, is congenital erythropoietic porphyria. Okay, so again, it's a really easy MCQ question. And then the other thing that they want you to know is that porphyrias can either happen in blood-producing tissues, such as the bone marrow, or they can have hepatic porphyrias, which are porphyrias that only happen in the liver, that are limited to hepatocytes. And like we talked about, you can have porphyrias that are hereditary, such as the AIP, acute intermittent porphyria, or Porphyria cutaneous tarda, or you can have acquired ones, such as poisoning. Specifically, lead poisoning was one of the examples that we gave, right? Lead poisoning can lead to porphyrias because it can also inhibit the enzymes. Okay, so let's go over the two really important conditions from this lecture. Acute intermittent porphyria. I'm already giving you the answer that it's a deficiency in the enzyme uroporphyrin synthase, right? You guys have to tell me, what would you expect to see in the labs? Elevated levels of what? In acute intermittent porphyria, where you lack the enzyme uroporphyrin 1, you would expect to see elevated levels of what? The answer is urinary ALA and PBG. The reason for this is because uroporphyrin synthase is responsible for synthesizing uroporphyrin from PBG. And so because the enzyme's not working, it leads to an accumulation of the substrates, specifically PBG, and then that can lead to an even more backup of ALA, right? So yeah, elevated levels of ALA and PBG are found in a deficiency in uroporphyrin synthase because the enzyme can't use these substrates to make uroporphyrin. Okay, for the next one, Porphyria Cutanea tarda is an enzyme deficiency in uroporphinogen decarboxylase, right? Try to remember the pathway in your head, uroporphinogen decarboxylase. What is this going to lead to? What urinary findings can I find? Think about it. Okay, and the answer is uroporphinogen or uroporphin 1 is going to be increased. Again, this makes sense. If I lack the enzyme that's responsible for metabolizing uroporphyrin, what that means is that I'm going to have an accumulation of uroporphyrin because it cannot be metabolized, right? The other thing, just as a revision of what I talked about before, acute intermittent porphyria is going to lead to abdominal pain and neuropsychiatric problems. And this is very, very different than the symptoms that you were going to see in Porphyria cutanea tarda, which is going to be skin manifestations, specifically photosensitivity. They're going to go out in the sunlight and they're going to present with blisters. Okay, so you guys tell me in acute intermittent porphyria, what enzyme is deficient? What is the deficient enzyme that we just talked about in acute intermittent porphyria? The answer is uroporphinogen 1 synthase. Okay. And because you cannot form heme in this scenario, how is this going to affect ALA synthase? Is ALA synthase activity going to be increased or decreased? The answer is that it increases, right? If I don't have heme, I no longer can inhibit ALA synthase. Because I'm not inhibiting ALA synthase, the activity of it increases. What is the pattern of inheritance for acute intermittent porphyria? What is the pattern of inheritance? Autosomal dominant, okay? And we already talked about the symptoms. It's neurological symptoms and also abdominal pain. Okay, so anxiety, paralysis, anything to do with the nervous system, neuropathy, motor problems, sensory problems, weakness. Then another clue that they might give you is that there's a port wine color. So the way that this happens is that the patient is going to go, they're going to urinate, and before they flush the toilet, right, if they let the urine sit in the toilet for a little bit, what happens is that the urine is actually going to change color to a darkish red. And so again, this only happens if the urine is left in the toilet for a little bit. It's not immediately as it's coming out of the body. And yeah, because I lack the enzyme uroporphinogen 1 synthase, I can't make uroporphinogen. It's an accumulation of the substrates, which specifically are ALA and PBG. Well, specifically it's PBG, but because you have an accumulation of PBG, that's ultimately going to lead to an accumulation of ALA as well. Okay, so we talked about Porphyria cutaneous tarda. I really emphasize the idea of photosensitivity. The patient is going to present that they went out into the sunlight, they left, and then it caused blister formation in Porphyria cutanea tarda. What is the enzyme that is deficient in Porphyria cutanea tarda? The answer is uroporphinogen decarboxylase. I cannot metabolize uroporphinogen, meaning there's going to be elevated levels of uroporphinogen because that is the substrate. And what is the pattern of inheritance in Porphyria cutanea tarda? The answer is autosomal dominant. We only have one autosomal recessive porphyria. Okay, so this is a clinical scenario that they might give you. As you can see, I highlighted the buzzwords. We have very sensitive to light. This is what we mean by photosensitivity. And we can see that after he went into light, he has recurrent blisters. And then we also see that he has the red bound tint if he did not flush the toilet. So all of these things in your head are making you think of Porphyria cutanea tarda. Another thing that they might give you a clue in is that drinking excessive alcohol, as you know, alcohol damages the liver. And so this is another thing that's also going to further exacerbate the porphyria. Okay, so now that we know what the scenario of Porphyria cutanea tarda, just tell me one more time, what is the name of the substrate that cannot be metabolized in Porphyria, Cutanea, Tarda. What is the name of that substrate? Uroporphinogen, right? And so there's going to be elevated levels of uroporphinogen in Porphyria, Cutanea, Tarda. Okay, so remember I talked about vitamin B6. Think back, what was the enzyme? What was the name of the enzyme that required vitamin B6? What was the name of that enzyme? Hint, it also was the rate limiting enzyme. It's called ALA synthase, okay? And so I don't think this is particularly high yield for you as first years, but just in case they test you on it, what they're talking about over here is that in cases of people with tuberculosis that were treated with isoniazid therapy, this isoniazid, right, which is the therapy for tuberculosis, can actually lead to vitamin B6 deficiency. Now follow this train of thought. If I have a vitamin B6 deficiency, that means that my ALA synthase enzyme can't be working as much anymore. And so it's going to lead to a porphyria. And once again, this is an acquired porphyria, right? And so essentially what happens is that I cannot make ALA. Because I cannot make ALA down the road, I can't make hemoglobin, or I can't make heme. If I don't have heme, I can't make hemoglobin. This leads to anemia. And the specific kind of anemia that this can cause is sideroblastic anemia. Okay, another example of a acquired porphyria. We already talked about this. What is the name of the enzyme that's responsible for incorporating Fe2+ into the porphyrin? What is the name of that enzyme? The enzyme is called ferrochelatase. The enzyme is called ferrochelatase, right? And the other thing that you really want to know about the ferrochelatase enzyme is that it can be inhibited by lead. Remember, I talked about that earlier on. The lead can inhibit the ferrochelatase enzyme as well as the ALA dehydratase enzyme. That's really important to know. And I'm sure you guys know this from your other lectures, but iron deficiency anemia is going to cause microcytic hyperchromic anemia. Okay, so in lead poisoning, what are the two enzymes that might be inhibited? I just mentioned this in the previous slide. What are the two enzymes that might be inhibited in lead poisoning? Ferrochelatase and ALA dehydratase. Just to remind you guys, lead poisoning, the main thing that should be thinking in your head in a clinical vignette is something to do with toys, like a child that's eating toys, or something to do with paint. And the child is going to present to the clinic with fatigue, abdominal pain, these sort of non-specific symptoms. Drop in IQ, insomnia, abdominal pain, like I talked about before. Okay, so let's do some application. We talked about vitamin B6. Try to think, is the delta ALA going to be increased or decreased in vitamin B6 deficiency? It's going to be decreased, right? In vitamin B6, it's a coenzyme for ALA synthase. And if I don't have ALA synthase, I can't synthesize ALA, therefore decreased levels of delta ALA. The other thing to know about vitamin B6 is that if someone is treated with isoniazid, which is the treatment for tuberculosis, this might lead to vitamin B6 deficiency and therefore might exacerbate or might lead to an acquired porphyria. Now let's look at lead poisoning. Try to think for a second, lead poisoning, how is that going to affect the delta ALA levels? Think about the enzymes that it inhibits for a second. And the answer is that it's going to increase delta ALA because lead poisoning is going to inhibit the enzyme ALA dehydratase. If you're inhibiting the metabolism of ALA, ALA is going to accumulate, and therefore you get elevated levels of delta ALA. And then just as a reminder, some of the things that you want to look out for in lead poisoning vignette is lead paint, pottery glaze, and sometimes again like toys and stuff like that. Okay, the very last thing that we're going to talk about, this is important because I have seen this tested on before, but remember we talked about before that the cytochrome P450 enzymes or the system, they contain heme inside of that. Remember that, right? So the cytochrome P450, they need to utilize heme. Okay, so I told you before that the cytochrome P450 system is responsible for breaking down certain drugs or stuff like that. An example of a certain class of drugs that need to be broken down by the cytochrome P450 system is called barbiturates. Okay, so barbiturates are a class of drugs that are metabolized by the cytochrome P450 system. Okay. And what that means is that the barbiturates are going to stimulate the cytochrome P450 synthesis, right? And because my cytochrome P450 enzymes are being more produced, and because heme is required to be produced in order to form cytochrome P450, that means that the heme levels inside of my body are going to be reduced because heme is being utilized to form the cytochrome P450. Now think about this. We've already talked about this. If I've decreased levels of heme, because the heme is being used to make cytochrome P450, what's going to happen is that if I have low levels of heme, now my ALA synthase is no longer inhibited, right? We talked about this before. We talked about how heme is an inhibitor of ALA synthase. So if I'm no longer inhibiting the ALA synthase, that's going to lead to increased activity of ALA synthase, right? And now if my patient has porphyria, right, this is going to lead to more precursors in the porphyrias. And that means that barbiturates are going to exacerbate the disease. So once again, just to follow the train of thought, I have barbiturates. Barbiturates are utilizing the cytochrome P450 system. Because they're utilizing the cytochrome P450 system, the cytochrome P450 enzymes are using heme. Because they're using heme, the heme is being depleted. Because the heme is being depleted, there's low levels of heme, which means decreased inhibition of ALA synthase. Because decreased inhibition, that means more activity of ALA synthase, means more precursors. More precursors, in the case of porphyria, means exacerbation of the disease or more disease severity. I just want to emphasize one thing that's so important. Listen closely to what I talked about of how heme affects ALA synthase. I said it decreases the inhibition. This is really important because sometimes the way that they're going to test you on this is they're going to ask you, does barbiturates activate ALA synthase? Does it increase its transcription? No, that's not what's happening. Barbiturates are going to lead to less levels of heme and decreased inhibition. Decreased inhibition is different from increased transcription. It's different from activation. Decreased inhibition of ALA synthase. And that's it for this lecture. I hope it was beneficial for you guys.